So I am super delighted today to introduce to you all Dr. Yasuhiro Arimura. He completed his PhD at the University of Tokyo, doing really great analysis on H2A-B and SEMP-A. And he worked also in like Dr. Karu Mizaka's lab, and now he's a stellar postdoc in the Funabiki lab doing a great work at Rockefeller. And I'm really excited to hear the work he has done on structural variations in different chromosomes. And without rambling too much, because I've been taking a bit long, I'm going to give the floor to Yasuhiro. Thank you so much for joining us.
Thank you very much. Thank you very much for the kind introduction. And I'm going to start my talk. And first, I'd like to thank the organizer for giving me the opportunity to share my research with the chromatin community. In this study, we have solved the first high-resolution nucleosome structure isolated from functional chromosome using xenopus egg extract system and Cryo-EM. As the audiences of this meeting knows well, the chromatin and nucleosome structure are very important for regulating many biological processes on genetic DNA. Using in vitro reconstituted nucleosome, some of these processes can be reproduced. And the structural mechanism behind those processes have been studied very well using the Cryo-EM or the X-ray crystallography. Meanwhile, there are many complicated biological processes that are not easily reproduced with in vitro reconstituted nucleosome, such as formation of the mitotic chromosome or heterochromatin and centromere. Since the structural analysis of the functional chromosome is only possible at low resolution at this point, the structural mechanism behind those processes are elusive. To solve this issue, we apply the xenopus egg extract system to the Cryo-EM. For those unfamiliar with the xenopus egg extract system, the xenopus egg are collected from the xenopus egg extract is collected from the xenopus egg crushed by the centrifugation, and the interphase nuclei are formed on the sperm DNA by mimicking the fertilization by adding sperm and calcium to the extract. And following this, by adding the mitotic extract, the cell cycle progresses to the M phase. The advantage of using xenopus egg extract is that both interphase and metaphase chromosome can be formed and easily isolated. And in this study, we started by focusing on mononucleosome to understand the structural unit of the interphase and metaphase chromosome. In addition to the iconic octameric structure, nucleosome structure, many other subtypes of the nucleosome have been reported. Nevertheless, the endogenous nucleosome structure in the chromosome had been unknown. And to purify the nucleosome from the xenopus egg extract chromosome, we cross-link the chromosome in the extract to keep the nucleosome structure, and then isolate it with sucrose cushion and digest the DNA to the mononucleosome sites with mNAs and correct the nucleosome fraction with sucrose gradient. And important to note here is that we isolated the interphase and metaphase nucleosome with the exact same method in order to do the fair structure comparison. With this method, we ended up getting 3 fractions, mono, di, origo, for each interphase and metaphase nucleosome. And this di and origo fraction consist of clustered mononucleosome containing many internucleosome cross-linking. And in the purified sample, the cell cycle specific protein and chromatin binding protein and histone modification exist. And using this sample, we perform the mass spec and Cryo-EM. And to assess the most abundant nucleosome structure in chromosome with Cryo-EM, we try to determine the structure as least biased as possible. For example, instead of selecting the 2D class based on their expected structure, we used the all high-quality 2D classes for the 3D structure analysis. And thanks for the great capability of the ab initio reconstruction of the CryoSpark software, we successfully determined 4 types of the cryo-EM structure simultaneously. Then we identified the protein that represent the cryo-EM structure with the help of the mass spec and structure prediction. And this is the nucleosome structure we determined. And both interphase shown with blue and metaphase shown with red were nicely overlapped with the crystal structure of the in vitro reconstituted nucleosome. This suggested that the major form of the nucleosome in the chromosome is the octameric left-handed form, and those structures are not largely changed during the cell cycle. This is personally a bit disappointing for me, but probably the good news for the people using the in vitro reconstituted nucleosome. And after that, to improve the resolution, we employed the machine learning-based particle picking software to parse. And at this improved resolution, the type of the amino acid residue can be distinguished by cryo-EM map. And to be honest, I've never expected that we could determine the nucleosome structure with this high resolution until I actually done it. Because it was reported that nucleotide positioning inside the nucleosome can be changed depending on the DNA sequence. In the chromosomal nucleosome, the DNA sequences are highly heterogeneous, and therefore I have expected that nucleosome structure may also become highly heterogeneous, and the resolution of the chromosomal nucleosome would be low. However, in reality, for both interphase and metaphase nucleosome, we could determine the high resolution structure, and also when you see the DNA local resolution of the DNA, it's actually not that bad. It suggested that the DNA sequence does not largely affect the nucleosome structure, and especially if you see along the superhelical location, 0 to 1 and 4, even the position of the phosphate group can be distinguishable. This suggested that the DNAs are highly stable and positioned along these regions independent from DNA sequence. And interestingly, many non-chromatin remodeling factors bind to the DNA less positioned region we found. So it is possible that they are evolved to target the region where DNA can easily remodel. And next, I will move on to the structure variation of the nucleosome. And for a better structure variation analysis, we try to keep as many nucleosome structure variant particles as possible during the cryo-EM processing. In the typical cryo-EM pipeline, 3D classification were performed with multiple nucleosome-like classes, and minor nucleosome structure variant particle assigned to the low-resolution classes may be removed. And instead, we use the multiple decoy classes dissimilar to the nucleosome structure to clean up the noise and non-nucleosome particle during the 3D classification. This procedure may keep the structure variation of the nucleosome to the final structure. And then, using this data, we did the 3D variability analysis using the CryoSpark software. We found the structure variation at the lesion of linker DNA and linker histone and H C-terminus tail and N-terminus tail and histone core particle. And after that, we focused on one by one on each structure variation and assessed them. And for today's talk, I'm only going to focus on the linker DNA and the linker histone. And as per linker histone, by comparing the interphase and metaphase nucleosome, we found that the extra density appeared around the nucleosome diet of the metaphase nucleosome when the linker DNA crossed. And for the better understanding, we isolated the 3D class that has this extra density using the 3D classification. And mass spec result showed that egg-specific linker histone variant H1.8 was enriched on the metaphase nucleosome. And to confirm if this density was H1.8, we made an atomic model of the H1.8 by homology modeling and fit it into the density. And as you can see here, the model was reasonably overlapped with the extra density. Therefore, we conclude that this density is H1.8. And this result reveals where H1 bind on the nucleosome in the metaphase chromosome. Previous structure study using in vitro reconstituted H1 bound nucleosome showed that H1 could bind to the various positions on the nucleosome. One of the positions is so-called on-diet position, and the other is the off-diet or the non-diet position. However, it was unknown which of the binding mode actually exists in the chromosome. Our H1 atomic model was nicely overlapped with the previously reported on-diet position, and it indicated that at least the on-diet binding mode of the H1 exists in the metaphase chromosome. Now I will move on to the linker DNA. And the linker DNA angle is the critical factor for determining the type of the chromatin folding. Recent study using Cryo-EM, super-resolution microscopy, and next-generation sequencing analysis have shown that the chromatin are partially folded to make the so-called nucleosome clutch. And the type of the folding inside this clutch can be changed depending on the cell cycle and depend on the genomic region. And thus far, many types of the chromatin folding have been reported, have been proposed. Importantly, both open and closed linker DNA nucleosome can form the compacted chromatin fold. And to understand the linker DNA structure feature in the chromosomal environment, as a control, we additionally prepare the nucleosome with the modified method that cross-link the nucleosome after releasing the nucleosome from the chromosome using the mRNAs. We call this nucleosome as freed nucleosome. Then we compare the structure variation with the nucleosome cross-linked at inside of chromosome. As a result, we notice that the linker DNAs of the nucleosome cross-linked in the chromosome are more closed and visible. This suggested that the linker DNA structure are changed when nucleosomes are in the chromosome. To confirm this, we performed another Cryo-EM calculation called in silico mixing classification. In this analysis, we first mix the same number of the particle from each sample and then perform the 3D classification using the 5 nucleosome map with the different linker DNA angle. After the 3D classification, we counted the population of the particle in each class. And this is the result of comparing the mononucleosome and oligonucleosome fractions. And for both mono and oligo fractions, the linker DNA of the mononucleosome of the interphase shown with blue and metaphase shown with red are more enriched to the closed linker DNA nucleosome compared to the free nucleosome shown with gray. This result suggested that nucleosome packing in the chromosome is not completely random. Instead, the linker DNA angle are restricted to the closed configuration in the chromosome. This supports the idea that the chromatin forms a partially ordered structure, and also our data suggested that the unit of those partially ordered chromatin might consist of closed linker DNA nucleosome. And this is the summary of this study. And from the average structure, we found that the majority of the nucleosome structure in chromosome are identical to the in vitro reconstituted nucleosome, independent from the DNA sequence, histone modification, and cell cycle. And from the structure variation, we found that the H1 bound at the nucleosome diet in the metaphase chromosome, and also the linker DNA tend to be in close configuration in the chromosome. Also, I couldn't talk about it today, but we've also observed that the two types of the structure variation of nucleosome core particle, and also found the structure variation in the histone tape. For those results, please check the preprint available by arXiv. Also, I'd like to suggest that the most important take-home message for this community might be the fact that high-resolution chromosomal nucleosome structure can be determined, even though the DNA sequence are highly heterogeneous. This means that now we can analyze the nucleosome structure found in the functional chromosome. And I am hoping that further update of this method enable researchers to capturing the chromatin structure during the complicated biological processes that are not easily reproduced with the in vitro reconstituted nucleosome. However, even if the method for analyzing those chromatin structure is established, it might be not enough to understand how those structures are formed and regulate their functions. To understand this, I believe that chromatin structure would need to be manipulated. With gene-specific extract system, chromatin structure can be changed using well-established technique like immunodepletion or inhibitor addition. Also, I'm currently trying to replace sperm with in vitro reconstituted chromatin to manipulate DNA sequence and epigenetic marks on the chromosome. Taking advantage of gene-specific extract system like this, I would like to understand how chromatin structure regulate their function. In closing, I would like to thank all the people who helped with this work, especially Hiro, my really supportive mentor, and Rochelle, who did cryo analysis of in vitro reconstituted nucleosome, and Ruby, who taught me the decoy classification method she established. And I also thank many pioneers in the chromatin and structure biology field. And thank you so much for your attention, and I'm happy to take any questions.
That was fantastic. That's great that we're able to see that from cells. I'm excited to hear about all the different types, like cell types you can then use, like different, like.
Yeah, it would be great to apply it to another. I can see one question.
Yes. In fact, I was going to ask, so Felix Muller-Planet had raised his hand. If you'd like to raise your hand again, feel free. Otherwise, I'm just going to ask your question here in the chat. Thank you for your question. He says, great. Oh, yes, raised his hand. Perfect. Okay, you should be allowed to.
Yeah, can you hear me? Yes, you can, I think, right?
Yes, I can hear you.
I'm just reading my own question that I just typed. I'm just reading it off from the screen. Great talk, thank you. Do you see any evidence in your data of subnucleosome particles?
Subnucleosome. I only found the hexosome with the control nucleosome. It's a freed nucleosome. I found the hexosome with this freed nucleosome fraction and with 3D classification, but I couldn't find it in that with the chromosome or nucleosome, the nucleosome crosslink inside the chromosome. So I assume that in the chromosome environment, nucleosome might be stabilized to not easily form the hexosome structure. Of course, probably those structure might be formed during as an intermediate, but probably the amount of those subnucleosome might be very small. Yeah, so that I think that's why I couldn't capture the structure.
And then, if I may, a follow-up question. So is the data quantitative in the sense that you can estimate the fraction of hexasomes compared to normal nucleosomes, or is it not quantitative enough?
I can count the particle to compare the number of the hexasome and nucleosome, but I'm not remember it. But yeah, I can count the number of particles.
Yeah. And your hunch is like 1%, 0.1%, 10%.
It's not more than 1%. If it's 1%, I cannot detect them. So it's probably between 5 to 10%. But I'm not remember the correct number. It would be included in the actual published paper in future.
Okay, thank you.
Great. Thank you. And then we have a couple of really interesting questions from Masahiro Nishimura. He says, great talk. Why do you think that the nucleosomes from the oligo fraction appeared in high resolution? And then the second question, which I'm also interested in, just curiosity, how many cells are required for such high resolution cryo-EM analysis? So we can go back to the first one. Why do you think that nucleosomes are from...
For the oligo fraction, it's kind of a simple answer. It's because oligo fraction can cluster. I can get more particle on the cryo-EM grid, and I can use more particle to do the cryo-EM. And somehow, if you use the mononucleosome, that nucleosome tend to be bind to the carbon support film on the cryo-EM grid, and the concentration of the mononucleosome get really low in the cryo-EM grid. So that's why the number I can count on the cryo-EM grid, EM grid, for the mononucleosome are really low. And yeah, even if I use the same concentration, the number I can use for the cryo-EM is a little different with oligo fraction. That's why oligo fraction can achieve high resolution. And in the second question, that in the case of Genpus egg extract, I use 8 ml egg extract for each condition. But if you use the cell culture cell or cell pellet, the concentration of the nucleus might be more higher than just say extraction. So I think it would definitely less than 8 ml, but yeah, I've never tested with other systems, so I definitely want to try it. That might be really great.
Cool. Yeah, so now we're having a bunch of questions trickling in. I think we only have time for one more, so I'm going to go ahead and ask the next question from Siavash Kurdistani. He also says, great talk. I think we can all agree, great talk. Can you briefly summarize what you found with the variant nucleosome?
Yes, for the variant nucleosome, it means that probably the nucleosome variation inside this nucleosome core particle, and with this the structure variation inside the nucleosome core particle, we found two types of the structure variation. One is we named the major structure variation, and other is the minor structure variation. And apparently, this major structure variation is and this major structure variation have sliding of the histone complex. It has more big movement, and this movement is somehow suppressed in the chromosomal situation, because if we compare the population with free nucleosome, that the amount of the larger major structure variation are small in the chromosome nucleosome. But in the case of minor structure variation, it is just the variation that has the tilting of the histone helix or other part, and those variations are not suppressed and exist in that chromosome nucleosome. And we found that in this minor structure variation, that the size of the acidic patch can be changed. So it means that probably during this minor structure variation, the protein binding on the nucleosome can be changed. And also we found that the hydrophobicity of the surface can be also changed. It could affect the nucleosome interaction. So yeah, that would be the summary.
Great. Thank you so much.